So let me change here something. So anybody can start sharing. Okay, Gabi, can you try sharing your screen?
Yes. Hello, I will try now.
Yeah. So Gabi is one of our PhD students at the institute, and she will tell you about SynergyAge.
Sorry. No, was it okay? I'm
It
not sure if
it was okay, but then it closed.
Oh.
Can you try again? Yes.
Do you see my slides? Do you see the presentation?
Yes, we do.
Okay. Hello, everyone. I'm Gabriela Bunu. I'm a PhD student at the Institute of Biochemistry. And today I will present you the database SynergyAge, a database for synergistic and antagonistic interactions of longevity-associated genes. As Robbie mentioned previously, there are more than 2,000 known longevity-associated genes, which are collected in the GenAge database. However, these studies only show how single gene interventions affect the lifespan. It's difficult to predict how these genes interact and what the lifespan of a double mutant will be, because the effect is rarely additive. When combining two genetic interventions, gene can be epistatic and then can interact in nonlinear ways. Also, data on epistasis is much harder to obtain than single gene intervention data, because we cannot do widescreen experimental studies. There are a lot of combinations that can be that can be tested, and it's not feasible to try them all in the lab. But fortunately, in the last 2 decades, there is a growing number of papers that report lifespan for double mutants, and in some cases, even triple or quadruple mutants. And in order to evaluate the synergism and antagonism of genes, we developed SynergyAge, a database with gene combinations that affect lifespan. The first thing we had to do was to define what synergism means in terms of lifespan effects. And here you can see the example for 2 genes. In order to classify the interactions between 2 genes, we need to know the lifespans for wild type, which on the graph is on the left side. It's considered the reference. Then we have both single mutants G1 and G2. On the plot, we show the difference in lifespan in percentages versus wild type, and the double mutant, which is represented as G1, G2. Individual effects are defined as I mentioned, as percentages compared to wild type. And when we talk about synergism, we compare the sum of individual effects, the sum of percentages of increase in lifespan for each single gene intervention, and the effect of the combination of the double mutant, so the increase in lifespan of the double mutant compared to wild type. By doing this comparison, we can define 5 types of interactions, as you can see in the examples on the right. Synergistic, when the combination has a greater effect than the sum of individual effects. Additive, when they are exactly the same or plus minus few percentages. Almost additive, dependent, when the effect is between the 2 individual effects. And finally, antagonistic, when the combination has an effect that is smaller than both effects. Here you can see actual examples for each type of interaction described before. In the first figure, in figure A, we see in C. elegans RSKS1 mutants that show only 20% increase in lifespan compared to wild type, and DAF2 mutants that have 168% increase. But the double mutant with interventions in both RSKS1 and DAF2 lives 4 times more than the wild type. IFE2 and LET363 are additive with regards to lifespan, as you can see in figure B. So the effect of the double intervention is equal to the sum of individual effects. Figure C shows an example of 2 dependent mice genes. Here I showed that we can define the same for negative effects too, so for short-lived mutants. So here the negative effect of the double mutant is greater than that of MGA-T5, but smaller than that of P10. In flies, SOD1 and cats act antagonistically. Even though decreasing the function of either of them increases the lifespan, the double mutant lives less than either of the 2 single mutants. In triple and quadruple mutants, the definitions get a bit more complicated. And for more than 2 genes, we defined 4 types of synergism. The most strict is the full synergism, if the lifespan change compared to wild type is greater than the sum of lifespan changes for any two intermediary K mutants and N-K mutants. For example, in figure A, the lifespan change in the triple mutant is greater than the sum of lifespan change in single mutant G1 and double mutant G2, G3. And this is true for the other two pairs of single double mutants. The most common definition for synergy is usually greater than the sum. So in this case, the lifespan change in the N mutant is only greater than the sum of the N lifespan effects. Figure C shows why this definition can be misleading. Here we have three interventions whose combined effect is greater than their sum, but the combination is not fully synergistic. There are two intermediary double mutants that have a greater lifespan effect than the triple combination. When we define this type of interaction dependent, and there is another example for this in figure E. However, in order to assess the full synergies for an N mutant, we need to know the lifespans for all the N factorial intermediary mutants. This is not a common experiment design, unfortunately, in the papers that we curated. So the data is incomplete in the sense of this definition. Often we only know the lifespans for some of the paths between wild type and mutant, and we define one path of mutants by the series of interventions used to construct the N mutant, starting from the wild type. So from the wild type to the single mutant, double mutant, and so on until triple or quadruple or whatever N mutant is in that paper. If all the known paths are monotonic, so if the lifespan increases with each sequential intervention, as in figure D, we consider the interaction partially known monotonically epistatic. The development of the database included many steps. After data mining of the scientific literature, the curated data needed processing to ensure the quality and the proper format for visualization. The web interface not only provides downloading and browsing data, but also a network view with various features. SynergyAge is a manually curated database which contains lifespan data for mutants with various interventions on at least 2 different genes. Besides lifespans, we also extracted and displayed on the website the experimental conditions, for example, temperature and diet. We curated data only from papers with experimentally validated results, where the N mutant was compared with the single gene mutants and preferably with a wild type as well, because we need to compute the effects of lifespan versus the wild type. Another validation step was to check if the survival curves were characteristic for a normal aging pattern. This step was needed to avoid cases when, for example, reduced lifespan was technical error sorry, or pathologies rather than a short-lived phenotype. So if the survival curve didn't meet the normal shape, then we discarded the experiment from the database. So far, the database has almost 7,000 lifespan values for more than 1,800 gene combinations. Most of the data come, of course, from studied in C. elegans, but the database also includes data for flies and mice. And in total, we processed almost 200 papers. We have curated more than 200 synergistic combinations, 2 additive and 120 almost additive. For better visualizing the available data, we built a web interface where the data can be viewed as a network. Each node here represents a mutant, and the edges are drawn from a mutant to a mutant with one more interventions, from wild type to a single mutant, from single mutant to double, and so on. The color green represents long-lived mutants, and the intensity is proportional with the average lifespan from all the papers in the database. And the biggest nodes are those that have which are the most studied, so for which we have many papers. This can be used as a tool to explore known data on lifespan synergies. The network also integrates data for KEK pathways and tissue expression. So the user can select all the genes that belong to a specific pathway, or the genes that belong to the same pathway or are expressed in the same tissue as the gene of interest. So if we select a gene, then there is a panel where we can select the KEK pathways, and for one of them, we can see all the other genes in the network that are involved in the same pathway or that are expressed in the same tissue. Searching the database for specific genes can be done either in the network view in the panel on the right, or by using the search option, which is available in another page. There are several filtering and sorting options: filtering by lifespan effect or by number of mutations, and sorting by mutant name or by change in average lifespan. The search query can include any number of genes separated by commas or semicolons, and the results are displayed in 4 categories. First of all, the exact match, if it exists in the database. Then every gene, which contains data for all the single mutants corresponding to the queried genes. And combinations that include at least one or all of the searched genes, but also other genes that were not searched for. And there is a toggle which can be used to switch between OR and AND. Each row summarizes the number of experiments, the range for change in average lifespan, and the type of epistasis for each mutant in the results list. For each N mutant, the website contains a page with detailed information on individual experiments and a graphical representation of all the reported effects. Nodes here represent individual cohorts, and edges between nodes are drawn only between animal populations from the same experiment. The rule here for edges is the same as on the previous network, from an N mutants to an N+1 mutant, and the shapes are also the same as in the big network. To visually evaluate the epistatic effect of a gene combination, user can click on an N mutant of interest and then use either select neighbors from the carousel panel, obtaining a list of N-1 and N+1 mutants, or using the button select genes with PubMeds and the ID, which will obtain all the mutants from the same paper. In the network view, you can see that more epistasis relationships appear for genes that are well studied. For example, DAF16 and DAF2, which are the central nodes in the previous network, are also among the most studied genes in C. elegans. This means that the number of interactions that we have in SynergyAge, or the genes involved in this, are somewhat biased by the existing literature. Unfortunately, this can affect to some degree the maximum effect that was observed and considered in our database. And this can be seen in this table, as more studied genes have more synergies. Still, considering this limitation, we wanted to analyze if any molecular connections could be inferred for each of the epistasis categories. Our analysis focused on identifying interactions between pathways. First of all, we identified longevity pathways by enrichment analysis for lags. After applying the multiple testing correction, 5 pathways showed enrichment at FDR less than 0.05. However, much more pathways contain longevity-associated genes. Considering all the pathways identified, we then computed the number of interactions inside each of them, and also the number of interactions which involve a gene from that pathway and another gene from another pathway. We noticed that there are synergies between genes that belong to pathways already known to modulate longevity, which you can see in the table on the slide. And it should be mentioned that synergies can be either completely part of a certain pathway, like you see in this slide, but also between different pathways. So as you can see, many synergies and dependent relationships also occur between distinct pathways. And again, all of these are related to longevity determination. Overall, this gives a broader view on how many synergies exist, but also how spread they are over basic processes and biological pathways. An interesting fact is that not all genes in SynergyAge are actually longevity genes themselves. Some genes have small effect on lifespan, which we define as less than 15%, but can be synergistic with a lag. We have termed these enhancers, and there are quite many of these in SynergyAge, about 10% of the total number of genes. And similarly to lags, they can also be found in some of the pathways mentioned before. To summarize, SynergyAge is a curated database for synergistic and antagonistic interactions of longevity-associated genes. It contains genetic and lifespan data for animal models obtained through multiple longevity modulating interventions. The website consists of an easy-to-use platform for browsing, searching, and filtering through the data, as well as network-based interactive module for visualization and analysis, and is a resource that provides information for a guided design on epistasis lifespan experiments. And the database is publicly available at synergyage.info. I would like to thank my colleagues and our collaborators that participated in this paper. The paper was published last year in Scientific Data. And of course, I would like to acknowledge the funding agency. Thank you. And if you have any questions, I'm happy to answer them.
Thank you very much, Gabi. An excellent presentation. It was very refreshing even for me. So thank you.
Thank you.
And if there are any questions, we can be late a few minutes. It's okay. I think I took a bit more time than needed in the beginning.
The same comment. Nice job.
Thank you.
Thank you, Vadim.
Very nice database. You know, in one of my papers I wrote, if the researcher would test each combination of longevity genes, so they will need the time comparable with evolution. So I think at least few million years will be more or less needed for such work, okay? There is a real need in such kind of databases, okay?
So just to add, maybe just to add a bit on the drive that made us start with this project. So of course we are interested in predicting synergistic interventions, and in order to do so, we need to start understanding about them, even if we manage to curate only a bit of the huge amount of potential combinations. Yeah. But
it's
yeah,
it's exactly what I wanted to say.
Yes, yes.
It's only in support. Okay.
We understand it's a huge amount of data, and actually we will probably have more difficulties maintaining the database because new papers appear all the time. So this requires a continued effort. Yes, but thank you for the comments, Vadim. Yes, thank you.
Thank you very much.
Any other questions?
Yeah, maybe just a short one. Could that be used to try to infer interaction more mechanistically? So here it's based on data, but have you seen some things giving some hints, for example, like, okay, that could be linked to a given gene regulatory pathway, gene regulatory network. These two genes, we know they should interact in some way. So actually we have no idea why they interact.
I have tried to predict effects based on the data that we have in SynergyAge. I have tried to predict based on distances in biogrid, pathways in which they are involved in ontology terms, and many other things. But unfortunately, I didn't manage to do that. The accuracy was low. But I guess that there is still not enough data, even though the database has few thousand entries. There is, first of all, a bit of bias because negative results are published less than the positive results. But still, there are a few genes that are more studied, a few pathways for which each researcher tests that the new genes in combinations with that. But we don't have genes about, we don't have data about all combinations of genes. So I have tried, but so far I didn't manage. In terms of database and website, it provides a few visualization tools, not analysis tools, but you can select various mutants or various nodes in the network and look at the pathways in which they are involved, or you can select genes in specific pathways. You can select multiple nodes, not just only one, and you can get a closer view to that part of the network and evaluate the interactions between them in that section. So I hope, I don't know, in a future update when we'll have more data to be able to predict or to explain interactions and effects between two or more combinations of genes, but unfortunately not right now.
Very nice. Yeah.
I would add something to this. So Gabi is very ambitious. So she took your question as, can we predict everything based on what we have? We do have a positive case, which will be presented in the afternoon. So we predicted a combination of genes, ODR3, IFE2, hopefully with DAF2 as well. And Ioan will tell us about the experiments that we carried out in C. elegans. But yes, what Gabi means is that it's not a clear pattern that we can predict from now on all the combination.
All right, just a few cases are already interesting. Really. Okay.
Yes.
Right.
Yes. So we had one success case so far.
Okay, very good.
Okay, cool.
Thanks.
So if there are no more questions, then we should probably move to a short coffee break.